Alright, so in this lecture, we're going to be talking about the general characteristics of the animal kingdom, and we're going to start talking about invertebrates. And those are our simpler animals, and we're going to work our way through the different phyla of invertebrates, and then after invertebrates, we're going to talk about vertebrates. So today, we're mainly going to focus, like I said, on the general characteristics. So this cladogram kind of shows kind of a breakdown of all the phylums or phyla of the animal kingdom, and our simplest, our sort of our out group here would be the sponge. So we go back here, and then you're going to see that that we become more complex. That we see the animals increase in complexity, and they increased in being able to live on land. Having lungs, having waterproof skin, and then here at the bottom, this will be our last group that we talk about. Will be the vertebrates. The fish, amphibians, reptiles, birds, and mammals. All right, so first of all, general characteristics of animals. So animals are made of eukaryotic cells. Remember that. That means cells with a nucleus and mitochondria and all those cell parts. They're multicellular. Animals are always multicellular, and they're heterotrophs. Remember, that means they cannot make their own food. We actually find that animals ingest their food in some way. No animal cells have cell walls. This is something that's unique to the animal kingdom. Pretty much every other kingdom we've talked about has at least some members with cell walls. Remember, fungi had cell walls of chitin, and plants had cell walls of cellulose. Animal cells do not have cell walls at all. We ingest or eat our food. Also, our life cycles and our embryonic development are different from some of the other groups. So we're not going to talk too much about that. I'll show you a quick slide of embryonic development here. So one of the things about animals is that our cells are always diploid, which means we specifically have 2 copies of every chromosome. This kind of goes back to genetics. And so as an adult, we're diploid. As a zygote, we're diploid. And all of this time, all of our cells are diploid through all of our life cycle. The only haploid cells that we have, remember those are cells with only 1 copy of each chromosome, are the sperm and the egg, which technically only live for maybe 24 hours. And if fertilization doesn't occur, then they just die off. So we don't have multicellular animals that have haploid cells. Our cells are all diploid, and that's different. Fungi are actually haploid. Plants actually have what's called an alternation of generations, so that's something that's unique. We also have a special embryonic development that occurs in all animals, and that's what this is showing, and we're not going to really get into that, but we do go through, the zygote goes through mitosis. It becomes something called a blastula and a gastrula, and it sort of folds in, and you get these 3 tissue layers, ectoderm, mesoderm, and endoderm, and each of those sort of become a different thing in your body. Ectoderm, for example, becomes the nervous system, it becomes the skin, etc. So there's some very unique things that happen. All animals develop some kind of a digestive tract and stuff like that.
Sea urchin development. Biologists have learned a lot about fertilization and embryonic development by observing and experimenting with sea urchins. Sea urchin eggs are fertilized externally after the animals release their gametes into the water. Fertilization and subsequent development are similar to the changes that occur in the early embryos of mammals, but they are much easier to observe in sea urchin embryos.
All right, so one of the things that we have looked at is the development of embryology. If you remember comparative embryology, and that tells us a lot about the relationships between different kinds of animals, but sea urchins are one that they've studied a lot. All right, a little bit of evolution. So the fossils of animals, you're not going to be tested on when the first animals appeared, but it was about 560 million years ago. The earliest animals actually were typically soft-bodied animals, and then later we see more animals with spikes and shells, and so these are just some pictures, but we're not really going to focus too much on the when and the how as far as the evolution. We're really going to focus more on the different groups of animals and just kind of looking at the different advanced and specialized kinds of structures that they have. Now, to understand that, you need to know that animals are organized into different phyla. That's the plural of phylum. And the phylum vertebrates we actually belong to. It's actually called chordata. And that's animals that have what in most of them becomes a backbone. Most animals, though, about 96% of the animals on Earth are actually invertebrates. There's way more of them than there are of the vertebrate animals. So, some other things that we need to talk about very quickly is, and this is, again, sort of looking at how animals have become more complex evolutionarily. Number one, the number of tissue layers. Remember, in an embryo, the embryo basically divides into these layers of tissue originally, and the simplest animals only have like 2 layers of tissue, the ectoderm and the endoderm. And then what we see is as animals become more advanced in their embryology, the embryo's development, we get ectoderm, mesoderm, and endoderm, an actual middle layer of tissue, which becomes muscle and a lot of the organs and things. Also, symmetry is going to be very important. So symmetry has to do with the body plan. And we'll talk, I think that the next slide actually has a picture of some of the different kinds of symmetry. Now, if an animal doesn't have symmetry at all, that's called being asymmetrical, or called asymmetry. And then there's 2 kinds of symmetry that we see in animals, radial and bilateral. Most of the more advanced animals have bilateral symmetry. There's also a special body cavity called a coelom. Looks like it should be coelom, but that's actually pronounced coelom. And the coelom, it's actually, if you were to cut a person's chest and stomach open, that big cavity where all the organs kind of neatly lay, that's the coelom. And the simplest organisms actually do not have that, but the more complex animals do. They have this body cavity that forms during embryological development in the middle of the mesoderm, and this is where all the organs can sit without being squashed. And then, finally, looking at embryonic development, there's 2 kinds of embryonic development. One's called protostome and one's called deuterostome. We'll talk about that in just a second. So this is a little bit about symmetry. So radial symmetry is where you could sort of, like, if you lay this down, think of like a pizza, and I could cut it any way, and no matter which way I cut it, the 2 sides would look the same. That's radial symmetry. Now, it's not the same. I can't cut it this way and get, necessarily, but it's from the, from the top. On the other hand, bilateral symmetry, there's a distinct, and you're probably going to need to know these words. So the front is going to be called the anterior. The back is going to be the posterior. The top is the dorsal side. And the bottom, the belly, is the ventral side. And so organisms that have bilateral symmetry, you can only cut them one way. If I cut them like this, I don't get 2 equal things, right? The 2 sides would not be the same as each other. So you can only cut it this way. So they literally have a top, a bottom, a left, and a right. Whereas here, although there is a top and a bottom, there really isn't, you can cut this any way you want to. So that's radial. There is another kind of symmetry called spherical, but there aren't any animals that have that. There's some protists that have that kind of symmetry. So that would be like where an organism looks like a ball. So I could pretty much cut it in any direction and it would be the same. But that's not true, with any animals. And then there's also the fact that there's just one group of the simplest animals that are completely asymmetrical. So they don't actually have any kind of symmetry. No body plan where one side mirrors the other. Now, as far as embryonic development goes, and that's what this is showing. So you start with a fertilized egg, that's the zygote, and then it goes through and it just makes a ball of cells. It becomes this hollow ball of cells called a blastula. And then that undergoes something called gastrulation, and it folds in. And we're not going to talk too much about this, but this is where, if you recall, in the most advanced animals, simple animals only get 2 layers of tissue, endoderm, ectoderm. So there's an outer layer and an inner layer when this folds in. The more complex animals actually get 3 layers of tissue. They get ectoderm, mesoderm, and endoderm. And they have there's 2 different embryological developments. Protostome, so stome means mouth, or technically it means opening, but it refers to the mouth, and proto means first. So if the organism develops mouth first, it's a protostome. And that's actually the less advanced. The simpler animals actually are protostomes. Deuterostome means mouth second. Deutero second, and stome has to do with the mouth. So what forms first? Actually the anus, the rear forms first. So when this folds in, the opening becomes the anus here. And that's actually the more advanced animals. Vertebrates are all deuterostomes. We go through this type of embryological development where the anus develops first and the mouth, the hole for the mouth, develops later. In the protostomes, the mouth develops first. And then the coelom, the body cavity they were talking about, which is called a coelom, you end up with ectoderm, mesoderm, and then this gap right here, this space, this is the coelom. So in that space, this is where we get all of our organs. So like our heart and our lungs and like your liver and all that sort of stuff that's found inside of our chest and our abdomen, that stuff all fits in that cavity called the coelom. And you'll actually be able to see that when we dissect a frog. That's actually where you'll really get to understand all the space that's in there, this big hollow space. The intestine actually ends up being lined by this endoderm. So the endoderm ends up being like the intestine, and that hole in the center is basically the intestine, going from the mouth to the anus in a one-way digestive tract. So your endoderm would surround that. Okay, so moving on. So we use this is how we build our phylogenetic trees, the phylogeny, or the evidence that supports like an evolutionary development. We use their body plan characteristics, we use the fossil record, when did these organisms live, because remember our current trees are built on evolution evidence, not just on what we look like. We look like some other organism, we don't put us related to them. We do it based on evolution. So we do it based on molecular data, like DNA sequences. We look at fossils, when did they live? We look at the body plan. We look at the embryology, all of that evidence we talked about in a previous chapter. And so you can see this is sort of the breakdown, and you can look at the time periods when these different things developed. And notice, so the sponges, no tissues, but then tissues developed. So all of these organisms now have tissues, and that was around 680 million years ago. And then that diverged, and we see that the symmetry, bilateral symmetry, radial symmetry. So the cnidarians, this is your jellyfish, they ended up with radial symmetry, and then these organisms all ended up with bilateral symmetry. And then that's around 670 million years ago. And then we see these sort of subdivide, and then we'll talk about that, which ones have a coelom, that extra body cavity, which ones have a one-way digestive tract, like they don't just eat their food and then it comes back out the same hole, like they literally have a mouth to an anus and it's a one-way tract through their body. So that's a development that makes organisms more complex. The development of a nervous system, the development of a brain. So we see that, we're going to actually look at that as we go through these different groups of animals. And this is just, again, another breakdown. Here's another one. This talks about the protostome versus the deuterostome. This is actually a really good one. So if you look here, you have no symmetry in the sponges. Then we have radial symmetry in the cnidarians, which is like your jellyfish. Then the bilateral symmetry comes about. Flatworms don't have a coelom. Roundworms have something called a pseudo or false coelom. Then the rest of these organisms have that special body cavity, the coelom. And then all the ones with the coelom, they have those three tissue layers, and they either have protostome development, which is where the mouth develops first, or deuterostome, which is where, remember, the anus actually develops first and the mouth develops second. And you'll see that the echinoderms and chordates, or like vertebrates, us, we are actually deuterostomes. And whereas all these other groups, the mollusks, annelids, and arthropods, are the protostomes. So we're going to stop with that, and then next lecture we're going to actually get into porifera, the sponges, and cnidarians, the jellyfish and sea urchins and whatnot, and we're going to go through their body plans and their special characteristics.